In this video, I'm going to be talking about two different ways that organisms can use sugar to make a form of energy called ATP. And the two different methods that organisms can use are cellular respiration and fermentation. Cellular respiration and fermentation are important in maintenance. You're constantly replenishing the cells in your body and you need ATP to do this. For growth, it's also important, as well as for activity. You're burning a lot of ATP and a lot of glucose whenever you are very active. Well, where does glucose come from? All the glucose that organisms use for energy actually comes from photosynthesis. So plants have special structures in their cells called chloroplasts, and these chloroplasts undergo photosynthesis and can produce glucose. Now, the interesting thing about glucose is that it's really good at storing a lot of energy, but our cells cannot use that energy directly. So in order to use the energy stored in glucose, our cells must convert glucose into another form of chemical energy called ATP. So ATP is necessary for a cell to actually do things. So when oxygen is available, organisms will typically break down glucose in a process called cellular respiration. Cellular respiration is also called aerobic respiration. Aerobic means it uses oxygen. And you could kind of think about this as if you've ever done any aerobic exercises. When you're doing aerobic exercises, you're using a lot of oxygen, and that's why they're called aerobic. So cellular respiration is an aerobic process, and it requires oxygen, and when oxygen is available, glucose will be broken down, and all of the carbon-carbon bonds in glucose will actually be broken down. So glucose is actually C6H12O6. This means that glucose has 6 carbon molecules in it. And those 6 carbon molecules those bonds get broken, so it releases actually 6 carbon dioxide molecules. So all those carbons in glucose get released as carbon dioxide. So when you eat food, you're eating all this glucose, you're breathing in oxygen, that oxygen is allowing you to break down the glucose, and all the carbons that were in the food that you ate are actually being breathed out in carbon dioxide. This process also makes water when you're breaking down glucose. And for every glucose molecule, your body can make up to 38 ATP molecules. So you can actually get a lot of energy out of one glucose molecule. You can get 38 units of energy. When your cells undergo cellular respiration, they take in glucose, and glucose begins to be broken down in the cytoplasm, that's the fluid in the cells, and then the rest of cellular respiration happens in the mitochondria. Remember that mitochondria kind of look like a kidney bean, and then they have all these folds in the inside. So in order for cellular respiration to happen, the mitochondria need to be involved in that process. If oxygen is not available, then cells will undergo what's called fermentation in order to get some energy out of glucose. Fermentation is known as an anaerobic process. Anaerobic. This prefix "an" means not. So that means it's not aerobic, so it does not use oxygen. Fermentation is not as efficient as cellular respiration. There's actually 2 different types of fermentation that are common in living organisms. One is called ethanol fermentation. Notice that glucose is broken down. There's some carbon dioxide that's released, and then ethanol is also created, which is an alcohol. But notice here that there's only 2 ATP molecules produced for every glucose molecule. So ethanol fermentation does not create a whole lot of ATP. What type of organisms undergo ethanol fermentation? Well, this would be things like yeast. And we use ethanol fermentation to make things like beer. That's how we get the alcohol in beer. Yeast is also used for baking bread. And we actually use the carbon dioxide that's released during fermentation to make bread rise. There is another type of fermentation called lactic acid fermentation. So the product of this type of fermentation is this large molecule called lactic acid. And notice here, only 2 ATP molecules are also produced compared to the 38 that you saw in cellular respiration. So both ethanol fermentation and lactic acid fermentation can obtain some ATP from glucose, but it's a very, very small amount. Lactic acid fermentation occurs in some bacteria cells, and also that occurs in our muscle cells whenever we deplete the oxygen reserves in our muscles. So to summarize, cellular respiration is the production of ATP by breaking down glucose using oxygen. So the inputs of cellular respiration are glucose and oxygen. If this is hard for you to remember, just remember this is what you are putting into your body. Your body uses cellular respiration for energy. So you're bringing in sugars, glucose, and what are you breathing in? You're breathing in oxygen. That oxygen is traveling to your cells, and that glucose is traveling to all the cells in your body where it's being used. And whenever that glucose is broken down, carbon dioxide is released. So that's why you breathe out carbon dioxide. And you also breathe out water vapors. So this water vapor and carbon dioxide that you are breathing out is produced all through this process of cellular respiration. When we talk about fermentation, this is the production of ATP by breaking down sugar in the absence of oxygen, also called anaerobic respiration. And in the case of ethanol fermentation, carbon dioxide would be produced as well as an alcohol called ethanol. Once again, notice there's only 2 ATP molecules produced for the 1 glucose molecule that goes into this process. Fermentation does not involve the mitochondria, and it actually occurs all in the cytoplasm of the cell. So how will you be measuring cellular respiration in our lab? You'll actually be measuring cellular respiration in plants. And remember that plants can make glucose, but plants also have to have mitochondria because they need to be able to convert that glucose into ATP. So plants use cellular respiration and photosynthesis for survival. Now, how will you be able to measure cellular respiration in plants? You'll actually be measuring it in germinating seeds. So here you can see a seed that's germinating, and germination just means it's growing. In order to get a seed to start germinating, what you do is you soak it in water for a couple of hours. So the seeds that you will be using will have been soaked in water for about 24 hours, and that actually signals the seed to start metabolizing and breaking down sugar stores in order to make ATP so it can start to grow. And the best thing about using a germinating seed is that it can't photosynthesize. So we don't have to worry about photosynthesis interrupting our measurements of gas exchange that we will be seeing. So let's look inside of a seed. Inside of a seed, it has a couple different parts. Out here is our seed coat. Notice that most of the seed is called the endosperm. The endosperm is just filled with starch, which is a type of sugar. So the embryo, the growing little embryo, will be breaking down this sugar and using it for cellular respiration in order to grow. How can we measure the amount of metabolic activity occurring in an organism? Or in other words, how can we measure how much cellular respiration is happening? Well, if you look at the equation for cellular respiration, it's really easy to measure these gases here. So you could actually measure how much oxygen is being consumed. The more oxygen being consumed, it means the more you're metabolizing. Or you could also measure the amount of carbon dioxide being produced. In our lab, you'll be measuring the amount of oxygen being consumed. Let me talk about how you will measure the amount of oxygen being consumed during lab. You will actually have 3 different conditions. These apparatuses are called volumeters because they can measure changes in the volume of gas that's occurring within each of these tubes. In one condition, you will have germinating seeds. In the other one, you will have dry peas and some glass beads in there. And then in the last treatment, you will just have glass beads. This will be your control. And you will be trying to hypothesize whether the germinating peas or the dry peas are going to be consuming the most amount of oxygen. The dry peas have not yet gone through the germination process, so they're kind of in a dormant state. Another thing that you're going to have in here on the bottom is this absorbent cotton. So you'll put some cotton swabs in there, and you'll also apply or drop on some potassium hydroxide. Now, why are you doing that? Potassium hydroxide actually takes carbon dioxide out of the air. So it's going to absorb any carbon dioxide that the peas release. So this actually allows you to only be measuring changes in oxygen in here, and any carbon dioxide that is produced by the peas will be taken out of the air and absorbed down here. So the carbon dioxide will not be affecting the changes in the volume in here. This allows you to have accurate measurements of just the oxygen. After you put your cotton swabs in here and your potassium hydroxide, then you'll put your peas or your glass beads in here, and then you'll put a stopper on the top and insert a pipette into the middle here. You will then lay down your pipettes in water here, and you'll let the tips of them sit out for a while because you actually want the inside of these tubes to equal the temperature in the water. So you're going to give it some time to equilibrate in there. Then after 5 minutes, you'll actually drop down your tubes. When you lower these pipette tips, you're actually submerging all three apparatuses under water. And what you'll notice immediately is a little bit of water will go into the pipette. And right away, you'll measure the distance from the tip of the pipette to that water. And that'll be your initial reading. Over time, what you may see, if, for example, the peas are consuming oxygen, the water will start to move into the pipette further and further because the gas volume in here is decreasing as the oxygen is being taken up. And later on, you will then measure again to see how far the water traveled in there after 20 minutes. And that'll give you an estimate of how much oxygen was consumed. The most difficult part about calculating the amount of oxygen that is consumed is actually correcting for any changes in volume that might have occurred just from changes in temperature and as well as maybe atmospheric pressure changes. So those type of things could have also caused the volume within these chambers here to change other than just, the amount of oxygen being consumed. So we need to correct for that. And we are going to actually use the control to correct for any changes that might have happened from factors other than cellular respiration. So in order to do this, first you need to calculate the net change of oxygen without doing any corrections. Let's say that our initial distance of water traveled from the tip was 50 millimeters. So that means, let's just pretend that the initial distance was here. So this here was 50 millimeters. After 20 minutes, let's say that it went 65 millimeters. So you can see the water actually moved in here, meaning the gas in here had to have decreased, the gas volume. So in this case, our net change of oxygen would be negative because gas was consumed, or oxygen was consumed within the chamber and the gas volume decreased, causing the water to move in. You want to do the same thing with the next treatment. What you notice here is that the volume didn't change at all, so I would put 0 down here. Well, let's look at what happened in the control. In the control, initially, the measurement that you would have gotten here was 62 millimeters. However, after 20 minutes, the distance from the tip was only 58 millimeters. That means the water had to have moved in this direction a little bit In order for that to have happened, the gas had to have expanded a little bit in the chamber, and that means that the net change in oxygen had to be positive. So you would put a +4 down for the control. So we need to correct for this because the control chamber shouldn't have changed at all, right? We just had glass beads in there. If the control chamber moved this way with just glass beads, that means something, maybe the temperature increased a little bit and caused the gas to expand. That means that probably happened in all of our chambers. So we'll have to subtract that change from each chamber because we want the change in the control to be 0 overall. So if I take +4, subtract 4, that makes 0. I also want to use my control to correct for all the other chambers. So now I'll take 0 minus 4, and that's -4. Take -15 minus 4, and that's -19. And this means then that with our correction, that the oxygen consumption in millimeters was 19 millimeters with the germinating seeds. In the dry soybeans, the net change in millimeters was -4, so some oxygen was also consumed in there, but not as much as in the germinating seeds, and the net change in the control with the correction was 0. So hopefully that helps you understand how to do these type of measurements, because you will have to do that in lab. What about fermentation? Let's talk about these two different types of fermentation. Remember, there's ethanol fermentation, which occurs in yeast. And remember, during ethanol fermentation, glucose is broken down, carbon dioxide is produced, ethanol is also produced, as well as 2 ATP molecules. Lactic acid fermentation occurs in animal muscles. And for example, again, remember, if our muscles deplete their oxygen reserves, then they will start using lactic acid fermentation in order to produce some energy and enough for us to maybe do a couple more reps or to sprint a little bit faster. Lactic acid fermentation also occurs in bacteria. What happens here again is glucose is converted into 2 molecules of lactic acid. Notice no carbon dioxide is made or anything like that in this process. Let's talk about some of the uses of ethanol fermentation. Remember that ethanol fermentation by yeast is used to make alcoholic beverages. We mix yeast in with barley, and then the yeast actually break down the sugars in barley, and we will use that ethanol they produce as the alcohol in our alcoholic beverages. If we mix yeast with different types of fruit, we can create wine and use the ethanol that they produce in those type of situations. Ethanol fermentation by yeast is also used to make bread rise. You mix the yeast in with the dough, and because as the yeast start breaking down the sugars, they actually produce carbon dioxide. And that carbon dioxide produced by the yeast causes the bread to rise. We also use ethanol fermentation in producing different types of biofuel. If you look at a lot of gas stations nowadays, a large percentage of the gas that you put into your car is actually ethanol, and that's created by yeast using ethanol fermentation to break down sugars in corn. Lactic acid fermentation is used in our muscles whenever the oxygen stores have been depleted, and it gives us that extra little boost of energy that we need. But lactic acid fermentation in bacteria is also used by us. When we mix bacteria with different dairy products, the bacteria break down the lactose sugar in the dairy products, and the lactic acid they produce causes the milk to curdle, and that's what gives us things like cheese or yogurt and sour cream as well. So we get very delicious foods by using the lactic acid fermentation that naturally happens in bacteria. In lab, you'll be measuring ethanol fermentation in yeast. Remember, yeast are single-cell fungi, so if you look at them under a microscope, each individual is just one single cell. And you will be mixing the yeast up in these tubes with different types of sugar to see which type of sugar they can break down easiest. You'll be testing glucose, lactose, sucrose, and as your control, water. After you mix the sugar with the yeast, you're going to incubate these tubes for about 40 to 60 minutes. And over time, you will notice that the more fermentation that occurs, the more carbon dioxide gas that will accumulate at this top of the tube. And this will actually start pushing the liquid on this side up to the end here. So which of these sugars do you think will be the easiest for yeast to ferment? Glucose is a monosaccharide, so it's just one single sugar molecule, whereas lactose and sucrose are disaccharides. If you want to look at how lactose is made, here is a single lactose molecule. Notice it's made up of 2 sugars. And the specific monosaccharides that make up lactose are galactose and glucose. So bind these 2 monosaccharides together and you get lactose. Sucrose, on the other hand, which is shown down here, is a combination of a glucose molecule and a fructose monosaccharide. So in order for yeast to ferment sucrose or lactose, they are going to have to first break the bond, the chemical bond holding these 2 monosaccharides together. So there is an extra step involved in breaking down a disaccharide versus just breaking down a monosaccharide, which they can just use immediately. So just think about that when you're making your prediction. After about 40 or 60 minutes, you will actually measure the amount of carbon dioxide gas that accumulates at the top of each tube to figure out which sugar the yeast was able to ferment the easiest. All right, so that is all the information that you need to know for the cellular respiration and fermentation lab.